Okay, I think well, we have 1 more minute, so I'll just wait for those who are not here yet. For those for those who signed in on Poll Everywhere, I know it might be late, but is there any way you could join the Facebook group? Because I think that might actually be more convenient. But if you can't, let's see what we'll do. It will be a bit difficult to use both formats at the same time. That's why I sent out that email last evening. Yes, Nick, you're watching the wrong one. We were done with lab. Look at the second lecture link. It should say bacterial genetics and organellar inheritance. That's the lecture for today. Okay, so you guys are coming in. I see Talissa. Okay, let me see here. Okay, so I'm just acknowledging those who are here, and then we would move forward. I think there's still some people coming in. Okay, I see Olivia. And Erica is here. I think we're almost done. Okay, Clemens, I got you. See you there. Alrighty. Okay, I think that's everyone I'm seeing. Let's see, how many do we have here? Let me go back. There. It's 9:15. Only have about 15. No, a little more than 15. 18. Let's see if anyone else is trying to get in. Okay, we'll just get started and Let's see, is there anyone else on? We'll go for it, see what happens. So, welcome. Good morning, everyone. It's 7:30 here, by the way. So we're finishing up our chapter on the bacterial genetics. Let me get to that. All right, so we're talking about the 3 modes of horizontal transfer. And so we talked about transformation, conjugation, and transduction. And yes, on Wednesday we talked about the products of different types of matings, conjugal matings. Can you tell me, someone, let's see if you could answer. I'd prefer if you answer on Facebook, but if you can't, go ahead and answer on Poll Everywhere. Yes, Clementine, I am getting your post. Okay, on Facebook. If you can't, here's the question: What is the product of a cross between an F minus and F plus bacteria? The product of a cross between F minus and F plus bacteria. That's the question. What's the product? And you could just add it to the comment list that we have there. Okay, Zach, you're saying F plus, and yeah, 2 F pluses. Great, great. All right, what is the product of a cross between, well, could someone tell me what is an HFR strain? What is an HFR strain? That's the second question. Okay, Erica, high frequency recombinant strain. What does that mean? I know that's that's what it stands for, but how is an HFR strain created? I should ask you. How is an HFR strain created? Can we just use the chat? Okay, we'll do the chat next time. Well, in a hurry. It's I don't want to switch things around at this point, Abraham. We'll use the chat next time. Could someone answer my question? How is an HFR strain created? Okay, great, Rayford, I got that. Okay, what is the product of a cross between an HFR and an F minus? Okay, great, Zach. Yes, HFR and F minus. Great. Anyone else? Let's see if anyone else answered well, the answer is there, so you could look at Zach's answers. But so yes, the product of a cross between an HFR and an F minus is an HFR and F minus. So there's no change to F plus. Okay, this one is a little bit more difficult. Why is there no change to the F minus strain? Why is it the F minus never becomes an F plus cell if the mating occurs between an HFR and an F minus? No, I prefer to ask the questions verbally, Nick, because it will take too much time to ask the questions if I have to type them. So I will ask verbally. Okay, so you don't know the answer to that question. So let's see what Zach says. It is because the F genome still remains in the F minus cell in addition to the fragment. Right, partially correct, Zach. The reason is that only a portion of the F plus genome of the F factor actually gets into the F minus cell. The entire F plasmid does not move over into the F minus cell when you have a cross between an HFR and an F minus strain. Hence, the product of a cross between HFR and F minus is that the F minus cell remains F minus, okay? So that is the reason for that. Then we talked about transduction, and we just began talking about generalized transduction. So I'm going to pull up the slides, and let's see how that goes. Okay. That's organellar inheritance. So where are we here? Okay, so we're looking at generalized transduction where any fragment of the donor, any fragment of the donor's chromosome could get incorporated into the head of a new phage particle. So here we have a phage infecting a bacterium that causes the bacterium to produce additional phage components of the phage particle. That's so the head and the other components of the phage particle. But it also results in digestion of the bacterial chromosome, the donor chromosome. When the new phage particles are being reassembled, some of the donor chromosome gets incorporated into the new phage particle. That phage that contains bacterial chromosome could then transfer this donor bacteria into a recipient, and thus you have another method of horizontal transfer from donor to recipient, but the transfer is mediated by a bacteriophage. So that's generalized transduction. You can use generalized transduction to map co-transduction frequencies, to map genes to know how close they are together on a chromosome. So for example, in this example, we have here a donor strain that is wild type for 3 markers: thymine, lysine, and cytosine. So this is the donor strain, and the recipient bacteria is auxotrophic for all 3. Transduction is allowed to occur, so using a phage known as P1 is allowed to infect this donor strain and incorporate the chromosomal DNA and then transfer that to the recipient strain. The next step is to select one of them, use one of the genes as a marker. So for example, if you use thymine as your selected marker, so you select for bacterial strains that are able to produce its own thymine, and you will do that by growing it on plates that lack thymine. So that way you select for thymine strains, and then you replica plate it onto plates that lack lysine or lack cytosine, and see whether or not there's growth. Or you could use lysine as your selected marker and then replica plate onto plates that are lack thymine and lack cysteine and observe where growth occurs. In this example, we see that strains that are positive for thymine, that is able to produce thymine, 47% of them are also able to synthesize lysine, and only 2% are able to synthesize cytosine. When lysine is the selected marker, you see that 50% of those lysine positive cells are also thymine positive, and none of them are cysteine positive. So this gives you an indication of the location of these genes on the chromosome. So here you see that the fact that lysine and thymine, right, lysine and thymine seems to transfer together more often, because if a strain is thymine positive, 47% of those would also be lysine. If a strain is lysine positive, 50% of those will also be thymine, but very little cytosine. So this indicates that cytosine is not transferring as readily with thymine or lysine. Hence, this data suggests that lysine and thymine are much closer on the chromosome than cysteine. So during transfer or during the transduction process, when the chromosome gets digested, because lysine and thymine are so close to each other, the frequency of them being transferred together or being co-transduced is greater than the frequency of either of them with cytosine being co-transduced. Okay, so let me check the Facebook to see if anyone's commenting or questions so far. Okay. Yes, the slides are on Moodle, Heaven. I posted it on Moodle. Okay, so you found it, great. All right. All right, so let's go back. Yeah. Okay, so the bacteriophages or phages can come in different types. We have what are called lytic or lytic phages or lysogenic phases, cycles that that bacteriophage can go through. So far, we focus mainly on the lytic cycle. So bacteriophage can either take part in a lysogenic cycle or the lytic cycle. And the lytic cycle occurs where the bacteriophage enters the bacterium, digests the bacterial chromosome, makes more phage particles, and then destroys the bacterium. That's the lytic cycle. In the lysogenic cycle, however, the phage DNA that infects the bacterium, instead of causing destruction of the bacterium, it integrates into the genome of the bacterium, similar to what happens with the HFR. So you have the phage DNA integrating into the bacterial chromosome. Bacteria that are able to phage particles that are able to integrate into the host genome are said to be temperate phages. So temperate phages have the ability of being either lytic or using the lytic cycle or the lysogenic cycle. Some phage are purely lytic, and some are purely lysogenic. But you have phage particles that could switch between either type depending on the environment that the bacterium is in. So if there's some environments that are conducive for, well, it's better off for the phage to stay in the genome, it would integrate into the bacterial chromosome. A phage particle phage DNA, I should say, that integrates into the bacterial genome is called a prophage. Okay, so the name we're just dealing with nomenclature here. The name of that DNA, that is the phage DNA that is inserted into or integrated into the bacterial chromosome, is called a prophage. Here's a very common one. Lambda bacteriophage is a very common temperate phage, so it could use both mechanisms. And so this figure just summarizes that. So here we have E. coli cell being infected with a temperate phage. It could either go through the lytic cycle and cause cell lysis, or it could integrate into the bacterial chromosome and creating what is called a prophage. Okay. At certain time, after lysogeny occurs, after the chromosome, after the phage chromosome integrates into the bacterial genome, forming a prophage, depending again on the environment, that prophage could excise out of the genome. It could just pop back out of the genome, undergo replication, and produce new phage particles and destroy the bacteria. So usually this lysogenic phase depends on the environment. When it becomes conducive to the phage particle, it will excise out of the genome. And it shows you here, when it excises out of the genome, so here we have a prophage here. That's a prophage that integrated into the bacterial chromosome. And it integrates at a specific site. Here is known as an attachment site. So you have an attachment site on the chromosome, an attachment site on the phage. These are similar in sequence. DNA sequences, the sequences are quite similar. So recombination occurs between these two and allow integration of the prophage. This prophage could excise out of the genome, and excision could be of two types. You could have normal excision. If normal excision occurs, then just the phage particle is excised out of the genome, and then it's replicated and new phage particles are produced. Or you could have what is known as abnormal excision. In abnormal excision, some of the bacterial genome gets excised and gets incorporated into the new phage chromosome. So you have abnormal or aberrant excision, and here you see that the galactose marker, which was originally on the bacterial chromosomal DNA, is now in the phage DNA. This type of this leads to, I should say, this abnormal excision could then lead to what is known as specialized transduction. If this phage particle that contains this galactose chromosomal DNA then infects a recipient bacterium, that process is known as specialized transduction. This is versus generalized transduction, where any gene could be incorporated and be transferred to the recipient strain. But with specialized transduction, only the genes that are adjacent to the phage insertion, so only the genes that are adjacent to the prophage, would get transferred to the recipient bacteria. This is called specialized transduction. Okay, I'm going to check to see if there are any questions. I don't see any. Oh, let's see here. Okay, I think we're good. No questions there. Oh, I just saw a bird. Okay, so I think the next slide just summarizes, summarizes a comparison between generalized and specialized transduction. Here we go. When the donor, so it says here, generalized transduction, this occurs during the lytic cycle. Specialized occurs when you transition from lysogenic to the lytic cycle. It says which donor DNA can be packaged with the phage. In generalized transduction, any bacterial gene could be packaged. With specialized transduction, only the genes near the integration site of the phage will get packaged. Okay, and I think that's all I want to cover for this chapter. That's all I will cover in this chapter. Okay. So I see here. Yeah, I'm gonna skip all the other slides. Okay, so that's it for chapter 13. Let's move to organellar DNA. Organellar inheritance, I'm sorry. Let me pull that slide up. Okay, so I know you know there's some FB that eukaryotic cells contain organelles, right? And there are two of these organelles, mitochondria and chloroplast, which you find in plants. These contain their own DNA. So far, we looked at nuclear DNA. We studied nuclear DNA. We looked at nuclear DNA in bacteria. Not bacteria, I'm sorry. We looked at nuclear DNA in eukaryotes. We looked at how it's organized. And we looked at how it's transferred. When Mendel did his studies really focused on nuclear DNA inheritance. But we know that these organelles, the mitochondria and the chloroplast, also contain organelles. So the question is, how are the genes in those organelles inherited? Is it similar to Mendel's? Do they follow Mendelian laws of inheritance? And the answer is no. But let's look at the genomes first, and then we'll look at how the genes in these genomes, in these mitochondria and chloroplast genomes are inherited. And this is actually showing you an example of organelle genome inheritance from chloroplast. The type of inheritance is known as maternal inheritance, and we'll talk some more about that. But we see this in the plants known as the 4 o'clocks. They have these green or variegated leaves, and we'll see how that comes about. Okay, so let's look at the mitochondria, the structure. We know the mitochondria it's a double membrane structure. It has a double membrane, an outer membrane, an inner membrane, and that inner membrane forms these convolutions known as cristae. And it's in here in the matrix, the inner part of the mitochondria is where you would find your DNA. The mitochondrial DNA. So let's look at the human mitochondrial genome. Very interesting. It's about 16.5 kilobases large, and it is circular, very similar to the prokaryotic structure. So it's a circular, double-stranded structure. The interesting thing is that there are no introns. No introns in the human mitochondrial genome. Shows you here how many genes, 37 genes, encoding tRNAs, ribosomal RNAs, and proteins necessary for oxidative phosphorylation. Because we know that mitochondria is the powerhouse. Mitochondria are the powerhouse of eukaryotic cells. That's where lots of ATP, or energy source, is produced. So and it does so, most of it, by a process known as oxidative phosphorylation. So a lot of the genes necessary for oxidative phosphorylation are found in the mitochondrial genome. So what you want to pay attention to is that there are no introns in the mitochondrial genome. In an organism known as a trypanosomes, they have an interesting type of mitochondria known as a kinetoplast. And this these trypanosomes have a single kinetoplast. Unlike humans, we have several mitochondria in our cells, but trypanosomes have a single mitochondria, and that mitochondria, the DNA, actually is arranged pretty interestingly. You have what are called mini circles and maxi circles. Mini circles and maxi circles. So you have the DNA, these mini circles and maxi circles forming these interlocking type of arrangement of the genome. The maxi circles produce most of the genes necessary for the kinetoplast function, and then you have mini circles, which are involved in what is known as RNA editing. So let's see here. Here's an example of RNA editing in kinetoplast. Now, RNA editing, there's no proof or no evidence of RNA editing occurring in human mitochondrial genomes. But in kinetoplast, which is kinetoplast again, is the mitochondrion found in trypanosomes, RNA editing is observed. Okay, so it says here, maxi circles contain genes, but the sequence does not exactly match the corresponding gene. The point here, here's what I want to take home here for this, that RNA editing occurs. It occurs in kinetoplast. And the mini circles, the genes in the mini circles are the ones that help in guiding this RNA editing. So here's an example of what happens. So here we have a pre-RNA, similar to a pre-RNA that we have in humans, right? So here's a pre-RNA. And what happens is that the mini circles produce what is known as a guide RNA. So this is the guide RNA. And the guide RNA helps to determine what the edited RNA would look like. So there's the editosome, which is a protein complex, uses the guide RNA as a template for editing the RNA, and so it shows you what happens here. So let's see. So this is your pre-RNA. The pre-RNA would align with the guide RNA. So G's would base pair with any uracil that is present. So G's could also base pair with cytosine. So you have GC base pairing, you have GU base pairing. These are all Watson-Crick type of base pairs. Okay. If you have, it says here, U's are inserted opposite of purines and any purines in the guide RNA. So if you have uracil present in the, here, let's see, uracils are inserted opposite purines. Okay. So here, so this is the guide, and these are purines. So uracils would be inserted into the guide, into the new RNA. Okay. So basically, the guide RNA is exactly what it says. It shows the pre-RNA, the adjustments that need to be made to produce this new type of RNA. Here you can see uracils being inserted. Again, uracils being inserted opposite any guanine or purine that is present. Okay. I think that's all I want to cover for there. Okay. The mitochondrial, there's some exceptions to the universal code. So we know what the universal genetic code is, but this is showing you that there are some exceptions to that. We know that UGA is typically a stop codon, but in mitochondria, it's tryptophan. Okay. Here another example. ATG is arginine in the universal code, but in mitochondria, it's a stop codon. You don't need to memorize this. The point is that there are, it says here, 5 differences between the universal and the mitochondrial genetic code. That's the take home for that. Okay, let's look at the chloroplast. So chloroplast, we know, found only in plants, and they are the where photosynthesis takes place. What I want to show you here with the chloroplast genome, it says that it's about 120 to 160 kilobase pairs long, but you can have more than one copy. of this genome in the chloroplast. But what I want to point out here is that chloroplasts actually contain introns. They do have introns. So that's the thing I want to point out here, is that we know that the mitochondrial genome lacks introns, but chloroplasts contain introns. That's what I want to mention there. Okay, let's see where I am here. I'm going to skip that. Okay, let's look at inheritance. Non-Mendelian inheritance of mitochondria and chloroplasts. So we know Mendel's laws of inheritance, and one of the key things we see here in chloroplasts and in chloroplast and mitochondrial inheritance, we see that the gender of the donating parent matters. Unlike Mendelian inheritance, the gender did not matter as to how the genes were transferred. But in organellar inheritance, it does play a role. Let me see, do I want to mention this? So just talking here about some of the mutations in mitochondria, what it causes. So mutations in mitochondria will lead to slow cell growth, and mutations in the chloroplast DNA could lead to decrease in chlorophyll, and that could change the color of the leaf, lead into leaf color change. So that's what I want to mention here. So let's look at inheritance. So we're looking at this is organelle. Let's see, is this organelle? No, this is actually mitochondrial DNA. So we're looking at mitochondrial inheritance, and it's a mutation in the mitochondrial gene called POKI. I don't know why. It's called POKI, and I'm showing you how it's inherited. So here we have a female, and a female with the mutation POKI, and a normal male. When a cross is made, the offspring of these all have the female or the maternal phenotype, okay? Maternal phenotype. But if the cross is between a normal female here, normal female, and mutant male, we see here again that the offspring all have the maternal phenotype. So in organelle inheritance, the inheritance may come from one parent. So all the contents of an offspring, all the genome contents of an offspring could derive from just one parent. Okay, we did not see that with Mendelian inheritance. Both parents contributed to the offspring, but in organelle DNA, that is not the case. It says here, when a POKI female mating cell was crossed with a wild-type male cell, all the spores were POKI. When a wild-type female mating was crossed with a POKI male mating cell, all the spores were wild-type. Okay, so this is an example of what is known as maternal inheritance. It's one parent doing the transfer, but it's the parent is typically female. So what are the mechanisms that lead to maternal inheritance? One is the size, the gamete size. The female gamete is much larger than the male gamete. So most, as a result, the offspring tend to have more of the maternal DNA, okay? It says zygote receives many female organelles and few paternal organelles, just because of the size. What happens, another reason, another mechanism for maternal inheritance is that the paternal organelles may be actively excluded or destroyed. That happens in plants, can happen in plants. It says another reason is that paternal organelles may segregate into non-embryonic organelles. So instead of becoming part of the embryo, they will segregate into non-embryonic cells. And another reason is that fertilization may prevent organelles from entering the egg. Okay, so these are all mechanisms that could lead to maternal inheritance. So here's an example of maternal inheritance displayed in these 4 o'clock plants. So you see this variegation occurring. Here we have a mixture of the white and green, okay, there, and all white branches here. So it's a variegated plants have green, white, and variegated branches. So green here, white, and variegated branches. So let's see what causes that. It shows you here the results of mating experiments with 4 o'clocks. This experiment was done by Carl Correns, and it shows you here. So mating between 2 green individuals, you end up with progeny that is green. But again, it depends on the phenotype of the donating female. Okay, so you see here, these are the phenotype of the females, and the phenotype of the progeny is always the what is donated by the female. Okay, so these are green and green and green. Here the progeny, they are all green. It doesn't matter what the pollen is contributing, what the male is contributing in this case. Okay, so we end up with that. Again, this is an example of maternal inheritance on display. So it says the plant is variegated because it has 2 types of chloroplasts. This is why we see the variegation. Okay, so it says wild type chloroplast DNA genes make chlorophyll. The mutant chloroplast DNA have mutations that prevent chlorophyll production. And so you end up with what are called, so here we have a cell. So cells in plants could have what are called heteroplasmic, could be either heteroplasmic or homoplasmic. Heteroplasmic cells contain a mixture of organelle genomes. So a heteroplasmic cell would have in their genomes, they would have mutant and wild type. Okay, so they have a mixture of organelle genomes, because you have to think in the chloroplast you have not just one DNA, right? You have several chloroplast DNA inside that chloroplast cell. And so you end up with, you can have cells that are heteroplasmic, that some of the chloroplast would have chlorophyll that has normal DNA and some with mutant DNA. So these are heteroplasmic. Homoplasmic cells contain only one type of organelle genome. So you could have, it could be homoplasmic for wild type, or it could be homoplasmic for the mutant. Okay, so that would be an example of a homoplasmic cell containing chloroplast with the same type of genome, but heteroplasmic cells would have a mixture. So here we have a heteroplasmic cell. Okay, you can see here the wild type chloroplast and mutant chloroplast. So that's heteroplasmic. Okay, when these undergo mitosis, so during mitosis, we know that the cells have to divide, and also the organelle genome divides. And when it divides, you could produce additional heteroplasmic cells here. Another heteroplasmic cell. But look at the extent of the heteroplasmy. You see here, this particular cell has much more mutant chloroplast than it have wild type chloroplast, as in this cell. So lots more wild type chloroplast. So upon further mitosis, you could end up with a cell that is homoplasmic. This one is heteroplasmic, but the intensity of the wild type is not as much. So you have less wild type cells here. Okay, this one you have much, it is heteroplasmic, but you have a lot more of the wild type genomes. And this one is homoplasmic for the wild type. So homoplasmic for wild type, homoplasmic for mutant, 2 heteroplasmic cells, but you have variations in the intensity of the heteroplasmy. And as a result, you end up with plants that have the variegation. So this is what would lead to this variegating pattern. You have portions of the plant that is all white, portions that will be all green, and then portions that are mixture, and the intensity of that mixture would change. So no two plants are going to look the same because of how mitosis could create these different levels or different intensity in heteroplasmy. Okay, I forgot to look at the slides. Let's see. Not the slides, I mean the Facebook. What are the TRNs in the human mitochondrial genome site? I don't know what you're asking here, Charlotte. I'm sorry. In the picture. What is TRN? I don't know what TRN is. We haven't gotten to that slide yet, Charlotte, so I think you're ahead of us. We will get there. All right. I think this just summarizes here. It says there's a threshold effect. A certain fraction of wild-type organelles is sufficient for making the plant phenotype normal. So here we have, let's look at this one, green. So this is a homoplasmic. You see all the leaves are green. This one would also be homoplasmic. All the leaves are white. And here we have some variegation, yeah? Here we have the mixture there. Here we have this portion is all homoplasmic, okay? So it's it says that there's a threshold effect. A certain fraction of wild-type organelles would be sufficient to make the plant have a normal phenotype, okay? So that varies. I will go to, let's look at mitochondrial diseases in humans. So it says here that several diseases in the human nervous system are caused by mutations in the mitochondrial genome, okay? And these mutations are passed from the mother to children, not from the father. And it says the symptoms could vary due to heteroplasmy. The intensity of those symptoms are dependent on, they vary, I should say. The intensity varies because of heteroplasmy. So here we have a mother with a mutation, normal father. And if you look at this pedigree, you'll see only when the mother has the mutation is the disease passed on, okay? So here we have a mother. She passes it on to all her offspring. Let's start with the first mother, the grandmother. She passes it on to all her offspring. Here, 1, 2, 3, 4. All 4 children have the disease, but only the females pass it on to their offspring. Here's a diseased male, and his children are all normal. Diseased female, all of them are diseased, and the diseased male, all children normal. So this is an example. This is a classic maternal or mitochondrial type inheritance seen in humans. Here's one particular one known as MRF, okay? MRF stands for myoclonic epilepsy and ragged red fiber disease, and you could read about the symptoms of it, but it's also it's associated with some mental retardation. But what this slide is showing you is that it's showing you how heteroplasmy affects the intensity of that of expression of this mutation. So here we have the female passing it on to all her offspring. Again, the male doesn't pass it on, and the difference in color is indicating the intensity of the percentage of the mutation, and that in itself affects the intensity of the disease. So here we have individuals with 94% of mutant DNA, and these have a lesser amount of mutant DNA, and of course the male, much less mutant DNA. Okay, he has 90% here. This individual has 73%. Okay, again, because of heteroplasmy. Well, how does that affect the phenotype? So it's showing you here individuals. I have 1 minute. Individuals that have 20% of the mutant DNA shows you which tissues are affected. So here the effect with only 20% of mutant DNA, mitochondrial DNA, it affects only the brain. okay? But as the percentage of mutant DNA, mitochondrial DNA increases, more tissues are affected. And that the tissues that are affected are determined by the extent to which that tissue needs oxygen or energy. Okay, so the higher the requirement, the higher the energy requirement for a particular tissue, the less it's able to tolerate mitochondrial mutation. So the brain has a very high requirement for energy, and so it's much more readily affected by mitochondrial mutations, because mitochondrial mutations affect the energy levels of the cell. Okay. And we will end on that note. Well, we're out of time. We'll finish this up on Monday and start the other chapter. I think it's on prokaryotic, what is it? Gene expression, prokaryotic gene expression. Okay, so I think that is it for today. Let's see where we are here. Charlotte, hey Charlotte, did I answer your question regarding the mitochondrial, the human mitochondrial genome? Oh, by the way, class is over. It's now 11:21, so we're done with class. But I just want to, if Charlotte, if you still need me to address your question, I could do that. Okay, but I will see you guys on Monday.